This is the part of the course where we're talking about how cell numbers are controlled. And in this particular lecture, we're talking about programmed cell death. Programmed cell death as it relates to cancer was actually my area of research throughout my PhD and my postdoctoral fellowship. So believe it or not, your cells actually have programs by which they can activate to cause their own death. And probably the most commonly studied, as well as the most common form of programmed cell death, is what we call apoptosis. A lot of students will initially think about cell death as being something bad, but you definitely need normal cell death. Programmed cell death is going to sculpt your fingers and your toes. During the embryonic stage, you have this webbing, and you need that cell death for that webbing to disappear. As well, programmed cell death will remove juvenile body parts. For a number of different species, they'll have large areas that undergo programmed cell death, say going from a tadpole to a frog. As well, you need programmed cell death to kill dangerous cells. If you recall from our previous lecture, you have MDM2 normally causing the degradation of p53. Let's imagine that a particular cell gets a mutation, gets an error where there's excessive MYC production. Well, if there's excessive MYC production, that's way too much S phase. The cell's going to keep on synthesizing its DNA. It's essentially going to replicate. It's going to get out of control. It might keep on dividing. It might form a tumor. It might form a cancer. As a backup mechanism, what you have is that with excessive MYC production, if there's way too much MYC, that actually activates a protein called ARF, A-R-F. ARF inactivates MDM2, and now you have stable active p53. And with stable active p53 can induce cell cycle arrest. If you recall from the previous lecture, this gives the cell a chance to fix the errors. If you cannot fix the errors, well then apoptosis, cell death, destroy that cell. Your body would rather have that particular cell be destroyed than have it replicate out of control and become a tumor or a cancer. So programmed cell death is definitely important. You need normal programmed cell death for health. As well, too much programmed cell death is of course really bad. If cells start dying all over the place, that's not good either. It's going to be about the right balance. Apoptosis, this programmed cell death, is highly regulated. It's really well controlled. And it's also stereotyped. There is a pattern to what happens in it. Necrosis is not really regulated, not really stereotyped. Necrosis is essentially cell damage. It's accidental cell death. So for example, with acute injury, cells can swell, they can burst into the surrounding tissue. With a particular cell, something else that can happen with necrosis, with cellular damage, is you can just break open the cell. You can destroy the cell. You can cut it. What happens with necrosis is that the inside stuff of the cell gets to the outside. And if the inside stuff of the cell gets to the outside, well, the neighboring cells are going to react. They're going to know, hey, that's not normal. Something bad's going on here. And essentially, the neighboring cells, the surrounding tissue, will undergo an inflammatory reaction. Just to mention it, inflammation is characterized by Latin terms rubor, tumor, calor, and dolor. And that's something you often hear on medical shows, but it's just a fancy way of describing inflammation. So rubor means redness, tumor means swelling, calor means heat, and dolor means pain. So you can imagine if you get a bee sting, you're going to have redness, you'll have swelling, you'll have some heat around the area, you'll have some pain, and that's inflammation. With apoptosis, you don't get that inflammation. The inside of the stuff does not get to the outside. Apoptosis is going to avoid the inflammation by very neatly shrinking the cell, collapsing the cytoskeleton, fragmenting the DNA, chopping up the DNA, right? The cell's destroying itself, so it's not going to need the DNA anymore, and actually signals macrophages, neighboring cells, for cell removal by engulfment. Neighboring cells are going to engulf the cell parts that are left over, membrane-bound compartments that are left over, and actually use those for whatever, for nutrients or something else. When you're reviewing this slide, make sure you go through three key terms. Apoptosis, that is that programmed cell death where the cell decides to destroy itself. Necrosis, that's when something else damages the cell. The cell didn't decide to destroy itself, something else damaged it. With necrosis, inside stuff gets to the outside, so the neighboring cells will undergo inflammation. Necrosis is not the same as inflammation, right? Necrosis, the particular cell got damaged. Inflammation is the neighboring cells going, oh, that's kind of messed up, right? The inside stuff is on the outside. Separately, you have apoptosis. Very neat, regulated, stereotyped, as in patterned program. So how is apoptosis triggered? There's actually specific proteins called caspases that trigger apoptosis. Caspases are cysteine proteases that cleave target proteins at specific aspartic acid residues. Importantly, caspases don't go around just chop random proteins. They go around and they chop specific proteins, and it's those specific proteins that might be inactivated or might be activated and that carry out the process of apoptosis. Caspases are synthesized as procaspase precursor molecules. Procaspases are cleaved and activated by other caspases, and this leads to an amplified cascade of caspase activity. Programmed cell death and apoptosis are often called cellular suicide. Now, I don't want anyone to think that I'm making light of suicide, right? Suicide is definitely, of course, a very serious issue. This is just how it is described in the scientific literature. So as you go on in science, you'll absolutely see people refer to apoptosis and programmed cell death technically as cellular suicide. Cell decides to destroy itself. If you think about how this occurs, it's really quite amazing in that all of your cells pretty much have these proteins, these procaspase precursor molecules in them, and all you need is to activate them and they'll cause a cell death. The proteins are already ready to go. And in fact, what happens is that procaspases are cleaved and activated by other caspases. So if you just activate some of these proteins, you actually get a chain reaction where you activate more and more caspases and you get more and more cleavage of these specific target proteins, some of which are inactivated, some of which are activated, but that lead to that cell death. So it's really quite fascinating to think that all of your cells can carry out this program by which they destroy themselves, and they have those proteins ready to go. They just need an activation. Let's look at how an individual caspase protein is activated. So we're going to look at procaspase activation, and it's done so by cleavage. There's going to be a little bit of variation between different caspases, but this is the general schematic. Here we have our inactive procaspases. As per usual with a standard polypeptide chain, you have an N-terminus and a C-terminus, N-terminus and a C-terminus. The polypeptide is actually divided into different domains. If you look in gray here, here's what we call a prodomain, and then you have this larger area and then this smaller area. What happens is that if you have an active caspase, well, one active caspase will activate other caspases, and how it does that is it goes to an inactive procaspase and essentially cuts at these particular cleavage sites. What then happens is the prodomains get discarded, and you have a large subunit getting together with a small subunit, with another small subunit, with another large subunit, and together they make an active caspase. That, of course, can go on to activate more caspases, also cut specific other target proteins, activating some, inactivating others, to cause these specific phenotypes of cell death. Notice that you go from two separate polypeptide chains to one multi-protein complex. Here we see the caspase amplification cascade. Your caspases can actually be further divided into what people call initiator caspases versus executioner caspases. Caspases are typically named after numbers. Your initiator caspases include caspase 8, caspase 9, and caspase 10. The executioner caspase is usually caspase 3. So what we typically see is the activation of some initiator caspases, 8, 9, or 10, and that will activate execution caspases, such as caspase 3. And there is definitely some feedback in that some executioner caspases will activate some initiator caspases, activating more executioner caspases, etc. As well, in many cases, initiator caspases will activate other initiator caspases. The executioner caspases, though, caspase 3 as an example, are going to cut the specific proteins that carry out the phenotypes of apoptosis. Some proteins it'll inactivate, some proteins it'll activate, and it's not a random process. One of the phenotypes of apoptosis, as we mentioned, is chopped up DNA. If the cell's destroying itself, it's not going to need its DNA anymore, so you're going to chop it up, stop any transcription. You can actually detect apoptosis by detecting fragmented DNA in a gel after the induction of apoptosis. Here you can see time, and you can see initially there's not really any apoptosis. The DNA fragments are quite large, and then about 3 hours later you start seeing a lot of this chopped up DNA, and by 12 hours there's a lot of chopped up DNA. Couple of important things here. First thing is that students will ask, well, how long does apoptosis typically take? Does it take 3 hours? Does it take 12 hours? It really depends on how strong the apoptotic stimulus is. In some cases, you can take less than 10 minutes and the cell will destroy itself. The second thing to note is that keep in mind the executioner caspases like caspase 3, they're not going to chop up DNA directly. Remember, executioner caspases, they're chopping up specific proteins, not DNA. So what typically happens is the executioner caspase chops up another protein, and then eventually you get the activation of some protein that goes to the nucleus to chop up DNA. If you know that apoptotic cells have chopped up DNA, well, you can actually use this to detect where apoptosis actually occurs, and the particular detection method that people use is called tunnel. So you can detect fragmented DNA in cells by tunnel labeling, and tunnel stands for terminal deoxynucleotidyl transferase mediated DUTP nick end labeling, which is a really fancy way of saying how you detect the chopped up DNA. So you can imagine if you have a developing limb here, if you have webbing here and these webbing cells are undergoing apoptosis, you'd expect to see chopped up DNA in them, and you'd expect to be able to detect them by what's called tunnel staining. So if you were to zoom up on that spot, you should see apoptotic cells. Apoptotic cells also change their cell surface properties. If you think about the cell membrane, and this side would be the extracellular space, this side is the cytosol, the lipids are not the same on both leaflets. In fact, the phospholipid phosphatidylserine in green is normally restricted to the inner leaflet of the plasma membrane, the side that faces the cytosol, shown for you here in green. In apoptotic cells, phosphatidylserine flips to the outer leaflet. And again, keep in mind, it's not like your executioner caspases bind to phosphatidylserine and flip it. No, they're going to cleave the specific proteins that eventually give you those apoptotic phenotypes. And why is this important? Well, in vivo, in the body, macrophages will recognize the exposed lipid. They'll recognize the phosphatidylserine that is facing the extracellular side, and they'll phagocytose the cell. So on the extracellular space side, you're going to have other cells like macrophages, and if they see the phosphatidylserine, they'll know to engulf the particular apoptotic cell. In experiments, probes for phosphatidylserine can detect apoptotic cells. With the idea that apoptosis is caused by an amplified cascade of caspase activity, starting with initiator caspases activating executioner caspases, the obvious question then is, well, what activates those caspase cascades? How do you get that first caspase being activated? Caspase cascades can be activated by either extrinsic or intrinsic signals. You have to be extremely careful here. A lot of students will think extrinsic means it has to come from the outside of the cell, and intrinsic means it has to come from the inside of the cell. That's not the case here. Extrinsic means via death receptors, and intrinsic means via cytochrome C. We'll see that again, but keep in the back of your mind, extrinsic doesn't mean it has to be outside, and intrinsic doesn't mean it has to be inside. That's not what we're talking about here. So let's first talk about extrinsic mechanisms. Here we trigger apoptosis kind of from outside the cell, but again, that's not the distinguishing feature. It's via cell death receptors. So for example, you have the killer lymphocyte activation of what are called FAST receptors. In the diagram here, you're going to see this killer lymphocyte essentially instructs this particular cell to undergo apoptosis. Maybe this is a damaged cell, maybe this is an infected cell, whatever. If it's unhealthy, this particular killer lymphocyte can tell it to undergo apoptosis. There's many different quote-unquote death ligands. One example of a death ligand is the FAST ligand. And the FAST ligand actually functions as a trimer. So it's drawn for you quite well here. The FAST ligand is going to activate the FAST death receptors. Just like there are many death ligands, there are many death receptors, the FAST death receptor being the particular one that the FAST ligand binds to. If you zoom up on the FAST death receptor, you can see that inside it has what are called death domains. FAST ligand binding to the FAST death receptor will activate its death domain, and the death domain on the FAST death receptor will bind to the death domain on the FAD protein. FAD is an adapter protein. FAD actually stands for FAST associated protein with a death domain. If the death domain on the FAST death receptor binds to the death domain on FAD, That then activates the death effector domain on FAD, and the death effector domain on FAD will bind to the death effector domain on procaspase 8 or 10, one of our initiator caspases. You do need death domain activation to activate the death effector domain. Notice the complex that you have formed, and this is what we call the DISC, as in death-inducing signaling complex. What we include in the DISC can actually vary. Notice here they've included your procaspase 8 or 10. They include the FAD protein. A lot of people will also include the FAS receptor. That's fine. Now what that does is that puts essentially the procaspase 8 or 10 in the right orientation to cross-activate one another. What's going to happen is that they will actually cleave one another. And if they cleave one another, what that gives you is an activated caspase 8 or 10. And if you have an activated initiator caspase, that will go on to cleave and activate a number of executioner caspases. They'll cleave the specific proteins again that give you the phenotypes of apoptosis. One thing to note is that the DISC, that particular complex, keeps on being activated, as in the complex doesn't have to hang on to the death effector domain and the pro domain from your previous caspase 8 or 10. It can get released. So a lot of students will ask, well, if the first two here activate one another, what happens to the third procaspase 8 or 10? Well, it can wait for a new procaspase 8 or 10 to bind to the other sections of the DISC or the other FAD proteins, and then they can cross-activate. So this whole cycle keeps on going, activating more caspase 8s and 10s, and then you get the activation of more executioner caspases. You can see you get an apoptotic cell here, and your killer lymphocyte can go activate apoptosis elsewhere. Caspase cascades can be activated by intrinsic signals as well. Here you trigger apoptosis via the release of the electron carrier protein cytochrome C from damaged mitochondria after cell stress. This is what it looks like. In the white you have the cytosol, you can see a mitochondrion here, and imagine there is a particular apoptotic stimulus. An example of an apoptotic stimulus that activates the intrinsic pathway is actually UV light. And this is why I want you to be careful. UV light obviously comes from the outside of the cell. It definitely doesn't come from within the cell, but UV light induces intrinsic apoptosis because it's actually going to affect the release of cytochrome C. So again, when you're thinking extrinsic, you're thinking death receptors. Intrinsic, you're thinking cytochrome C release. Intrinsic apoptosis can be induced by something from the outside of the cell. Cytochrome C is normally in the intermembrane space, but with an apoptotic stimulus, it gets released into the cytosol. In the cytosol, what happens is it will activate APAF1. Here is the APAF1 protein. ATP is involved in the process as well. Notice APAF1 has this card domain, and what happens is that multiple APAF1s, each with their specific card domain and binding to cytochrome C, will form this large structure. Very often this is described as a pinwheel structure, kind of like spokes on a bicycle wheel. And this is a structure that's often called the apoptosome. This structure can then recruit and activate procaspase 9. Notice procaspase 9 has a card domain, has the pro domain, and then the different units here. And you can see it does assemble this really large structure. Now, one thing to note is that some people will call this the apoptosome. Some people will call this thing the apoptosome with the procaspase 9. This huge structure is then thought to use its caspase 9s to cleave and thereby activate executioner caspases. And that, of course, leads to apoptosis. One thing that's quite interesting about this structure is that it is also thought that procaspase 9 may not get cleaved and then may not have the standard structure of, say, a large subunit with a small subunit with a small subunit with another large subunit. The apoptosome just might function together like this to cleave a lot of caspase 3s. Here we see an experimental view of intrinsic apoptosis. In the control case, you can see cytochrome C staining here. It's linked to a GFP. So you can see the green spots are essentially cytochrome C. And if you stain these cells with a mitochondrial dye, you can see that the mitochondria, well, that's exactly where the cytochrome C is. That's where you expect under control case cytochrome C in mitochondria. If you treat cells with UV light, specifically the cells that are circled for you here, you can see the cytochrome C is no longer only in the mitochondria. The cytochrome C is in fact everywhere in the cell. It's been released to the cytosol. You can also use here an anti-cytochrome C antibody, and with the anti-cytochrome C antibody, you can see that, in fact, yes, cytochrome C is in the cytosol spread throughout. So again, something external like UV light can induce intrinsic apoptosis. Whether it is extrinsic or intrinsic apoptosis, a common mechanism is that signals lead to the aggregation of the top caspase, promoting self-cleavage. So either you're going to activate a caspase 8, 9, or 10 to eventually get lots of caspase 3 activation. Many cells make inhibitors of apoptosis. This increases the threshold for activating the apoptotic program. Extracellular inhibitors include things like decoy receptors. Decoy receptors act by competitive inhibition. They might have something like a ligand binding domain, but not a death domain. And what this does is this outcompetes functional fast death receptors for ligands. As an example, if you have a particular, say, killer T lymphocyte moving around the body with its fast ligand, it's going to bump into a lot of cells. And you can't just randomly cause cell death whenever you bump into a neighboring cell. That'd be really bad for your body. So what healthy cells do is they actually make a lot of these decoy receptors. If you look at the decoy receptors, they'll be able to bind to the fast ligand, but the decoy receptors are actually missing the death domain. So what happens is if a fast ligand bumps into this healthy cell, well, you're probably just going to bind to a decoy receptor. You're not going to get any activation inside. And so for a healthy cell, it might make tons and tons of decoy receptor, but very little fast death receptor. So it's very, very, very unlikely that if a killer T lymphocyte bumps into you, that this would just randomly undergo apoptosis. So why isn't the apoptotic signal sent in this particular case? Well, the decoy receptors are missing the death domain. There are intracellular inhibitors of apoptosis as well. Some do competitive inhibition. For example, some might mimic an initiator caspase, but lack a proteolytic domain. Other inhibitors of apoptosis simply block the apoptotic machinery. BCL2 is one of these proteins that blocks the apoptotic machinery, and it inhibits channel formation in the outer mitochondrial membrane. Imagine in white here we have the cytosol. Here's a particular mitochondrion with a particular apoptotic stimulus that induces intrinsic apoptosis. Cytochrome C is going to be released from the intermembrane space to the cytosol. How that works is actually there's a number of different proteins called BIO230 proteins. It's a family of proteins. With the particular apoptotic stimulus, the BIO230 proteins will aggregate. They'll get together and essentially form a channel, allow cytochrome C to exit and go into the cytosol. Do notice that in addition to cytochrome C in the intramembrane space, there are some other proteins. These other proteins will leave and go to the cytosol too. They're quite important. We'll talk about them in a sec. BCL2 protein will prevent BH123 proteins from essentially aggregating. It'll prevent them from allowing the release of cytochrome C and the other intermembrane space proteins. So BCL2 essentially inhibits intrinsic apoptosis. I'll mention it here, but we'll see this in the next lecture, in that a lot of cancers actually have too much BCL2 or too much proteins that resemble BCL2, and what happens is that they are resistant to apoptosis. It's much harder to kill these cancer cells. Sometimes a lot of BCL2 and BCL2-like proteins allows them to be resistant to treatment. There are also proteins called IAPs, or inhibitors of apoptosis, and they block caspase activity in the cytoplasm. Imagine in white we have the cytoplasm, and remember what we said at the beginning is that in your cells you have these procaspases, they're ready to go. What can happen every once in a while though is that these procaspases are diffusing around the cytosol, they have some energy. Every once in a while, by mistake, two procaspases are going to bump into each other at the wrong place, at the wrong time, with the wrong energy, and you'll get spontaneous activation. Obviously, your cell has to prevent this from happening. It can't just randomly undergo cell death just because two caspases happen to bump into each other and cleave each other. You don't want that amplified caspase activity to just randomly start. So what happens is that your cell actually makes these IAPs, these inhibitors of apoptosis. And in a lot of cells, they'll have these IAPs drifting around the cytosol, preventing any caspase activity. IAPs are great at preventing caspase activity, but if you want to actually produce apoptosis, if you want the cell to undergo cell death, well, you're going to have to stop the IAPs. And how this works is if there's a particular apoptotic stimulus that activates the intrinsic pathway, well, of course, it's going to get the BIO230 proteins to aggregate. They're going to allow cytochrome C to release from the intermembrane space to the cytosol. Cytochrome C, as we know, will bind to APAF1, cause the apoptosome to form. Here we include caspase 9 with the apoptosome. That's going to activate executioner caspases. And to stop the IAPs, what you have coming out of the intermembrane space are other proteins, including anti-IAPs. So the anti-IAPs block the IAPs, they block the inhibitors of apoptosis. Now apoptosis can occur. Signaling can either induce or prevent apoptosis. In populations of dividing or stable cells, signals can induce apoptosis. After cell populations fully grow, apoptosis often becomes the default state, and signals are required to prevent apoptosis. The default state kills off cell if they leave their protective environment. Survival factors are used to control proper cell numbers in body tissues. Survival factors act through apoptotic inhibitors to protect cells from apoptosis. In this diagram, we're looking at 3 examples of how survival factors function. So you see survival factor here, survival factor here, survival factor here. One thing to be careful of is what they show at the bottom, because the textbook actually does not draw this for you quite perfectly. At the bottom here, it says apoptosis blocked, but notice there's an inhibition sign. So if you think about it, it kind of looks like inhibiting apoptosis block. So is that a double negative? Does it cause apoptosis? No, that's just not drawn for you well, in that if you're looking at survival factors, obviously at the end of the day, you should block apoptosis. You should block cell death. So very quickly, let's take you through these with a particular survival factor. One example is where you activate a particular receptor. Eventually, you activate a particular gene regulatory protein. This goes into the nucleus, causes transcription. You get translation in the cytosol. And if this particular gene that gets transcribed and eventually translated is BCL2, we know BCL2 is going to block apoptosis. That's how some survival factors work. Another example is survival factors may activate a particular receptor, which eventually leads to the phosphorylation of AKT kinase. AKT kinase is active when it's phosphorylated. Now, check out what's happening here. BAD normally stops BCL2, but if AKT phosphorylates BAD, it actually inactivates BAD, then BCL2 will be activated. And if BCL2 is activated, you're going to block apoptosis. So if you notice how this particular protein was named, it's kind of misleading in that active BAD stops BCL2. So BAD, if you have a lot of it, actually helps apoptosis go, as in a lot of BAD would cause cell death. However, apoptosis isn't always bad. If you think about something like cancer cells, it would be great to have apoptosis. It would be great to have a lot of bad protein in that. As well, in unhealthy cells, in certain cells during development where you want apoptosis, you actually need BAD. In the final example, you can see a survival factor activates a particular receptor. Eventually, you activate a MAP kinase. Check out how HID normally works. HID normally stops IAPs, and if MAP kinases are activated, they'll phosphorylate HID, making HID inactive. And if HID is inactive, you can't stop the IAPs, so the IAPs do their thing. They inhibit apoptosis. Determining neuronal cell numbers actually occurs very often via competition for survival factors secreted by target cells. During development, you actually have a lot of nerve cells, and what happens is particular target cells will secrete survival factors. And the neurons that get enough of these survival factors will survive, whereas the ones that don't get enough essentially will undergo apoptosis. So cell death adjusts the number of nerve cells to the size of the target. If you think about the overall picture, signaling between cells in a tissue allows the tissue to regulate its size. Imagine in the square here, we have cell A secreting a particular survival factor that might activate a particular receptor on a neighboring cell, and this cell, cell C, would be protected from apoptosis. Instead, with another cell, maybe it secretes a mitogen that activates a receptor and activates a pathway, and here cell D is told to divide, protecting cells from apoptosis, causing cell division, causing apoptosis. These are all methods to regulate tissue size. As an example, billions of cells die in our bone marrow and intestine each hour. The system is regulated so that cell division equals cell death.